Welcome to Biot Anime. HSP-70. Members of the HSP-70 family are strongly upregulated by heat stress and toxic chemicals, particularly heavy metals such as arsenic, cadmium, copper, mercury, etc. The HSP-70 chaperone system is involved in 1. The import of proteins into cellular compartments such as mitochondria, the endoplasmic reticulum and the Escherichia coli periplasm. 2. Folding of proteins in mitochondria and of a subset of proteins in the endoplasmic reticulum and the cytosol. 3. Disassembly of protein complexes such as the nucleoprotein complex, which initiates replication of bacteriophage lambda DNA in the chaperone baskets of chaperone-coated vesicles. 4. Degradation of unstable proteins and the bacterial heat shock transcription factor. 5. Control of the activity of regulatory proteins such as heat shock transcription factors and the initiator protein of plasmid replication. The members of HSP-70 family include DNAK, HSCA or HSC66, and HSCC or HSC62. In prokaryotes, the eukaryotic organisms express HSC-70 in cytosol, HSP-70 and its paralogs HSPA1A, HSPA1B, and HSPA1L, binding immunoglobulin protein BIP or GRP78 in endoplasmic reticulum and MTHSP-70 or GRP75 in mitochondria. The HSP-70 chaperone system constitutes other essential factors, co-chaperones such as HSP-40, J proteins and HIP, which aid in HSP-70 function, and nucleotide exchange factors. HSP-70 class of chaperone contains two major domains: N-terminal ATPase domain that binds to ATP and hydrolyzes it to ADP. The exchange of ATP drives conformational changes in the other two domains. The second domain is the substrate binding domain or SBD, that can interact and transiently associate with short linear peptide segments of folding intermediates. The SBD can be further subdivided into substrate binding subdomain and AC terminal subdomain. The substrate binding subdomain contains a substrate binding pocket that has a high affinity for neutral, hydrophobic amino acid residues. The C-terminal subdomain is rich in alpha helical structure, acts as a lid for the substrate binding pocket. When an HSP-70 protein is ATP bound, the lid is open and peptides bind and release relatively rapidly. When HSP-70 proteins are ADP bound, the lid is closed, and peptides are tightly bound to the substrate binding domain. All the cellular functions of the HSP-70 family are attributed to the ability of these proteins to bind to exposed hydrophobic segments in the substrate proteins or partially folded polypeptide chains, preventing non-productive association of the hydrophobic regions and thus facilitating proper folding. The intrinsic ATPase activity of HSP-70s is very weak for stoichiometric coupling to the substrate binding release cycle. And co-chaperones like J proteins and nucleotide exchange factors function as accessory proteins, required for facilitating this process. Interaction with J proteins, which catalyze the ATP to ADP conversion, causes the conformational change, is responsible for stabilization of the substrate-bound form. Furthermore, J proteins are believed to bind protein substrates and recruit them to HSP-70 at this stage. Thank you for watching the video. For more videos, please subscribe to this channel and press bell icon. See you again in the next video.